friends, welcome to our show, MS show. MS as in M stands for Madhu and S stands for
Sreeja.
my wife. And we are both doctors. And this show, the idea behind this show is that we want to explore the hidden realities behind important topics, explore beyond what is visible on the surface, dig deeper into certain questions, certain important questions of our day, important things which we find that which has not been explained very well in the usual media. So we want to explore that further. So the present topic is the coronavirus, and so first let us see what exactly is a virus. So I'll explain for my non-doctor friends or people who are not scientists, what exactly is this virus? So a virus is it's basically a strand of DNA or RNA wrapped in a protein coat. So we have the same thing, DNA and RNA, inside our bodies. That is the genetic material. That is the basic genetic material. So the virus is this genetic material which is wrapped inside a coat of protein. And some scientists say that it is living in the gray zone between the living and the non-living. And so what does this virus basically do? What it does is, it attacks our body, and inside our body, our cells, our human cells, they make the proteins which are necessary for our normal bodily functions. And these viruses, they attack these cells, and inside this cell is a center called the nucleus. So these viruses, they take over the nucleus and makes proteins, it makes the viral proteins. So the cell starts to make the viral proteins instead of the
over entire cell machinery.
Yeah, entire cell machinery, and it makes copies of itself, divides and makes many copies of itself. So basically our cells are now making the viral proteins, and the cells are making multiple copies of that virus, and so there is an exponential increase of virus in the body which will cause disease. So next
we will see the details of coronavirus. When we talk about coronavirus, we all think that it is actually a single dangerous virus. But corona means it is actually a family name of a large number of viruses, which is having the RNA as the genetic material.
So within the viruses, there is one particular family called coronaviruses.
The word corona comes from the spikes on the outer layer of these viruses.
Yeah, it's actually a Latin word, and corona means like the rays of the sun are spreading. So that corona stands for those rays of the sun. And so you see all the coronaviruses, you will see the spikes on them.
There are actually 6 main types of coronaviruses, and most of these viruses are not dangerous at all and will not cause any diseases in man. But rarely there are some coronaviruses like SARS, SARS, severe acute respiratory syndrome, which cause deadly diseases in human beings. And this SARS has got 2 varieties, SARS coronavirus and SARS coronavirus 2. And this SARS coronavirus 2 is causing COVID-19 infection.
Right. And so let's see this SARS pandemic which happened in 2003 to 2004. This SARS virus which you talked about, it actually caused a pandemic in 2003 to 2004. I think 2002 to 2004 time. And so let's first see what exactly is a pandemic. Pandemic is basically a widespread disease. That is, it's a disease which is happening on multiple continents, at least 2 to 3 continents at the same time. And so this SARS pandemic was also a respiratory infectious disease, and so they had isolated this SARS virus, which is this coronavirus which we talked earlier, as the infectious agent for this pandemic. And actually you'll be interested to know that this SARS virus, SARS-CoV-1, the first SARS virus, had a much higher mortality compared to the present coronavirus. So actually, when we studied this topic, the SARS virus had at least 10 times more killing power than this present virus. The mortality was 10 times higher. But there was so actually this and the SARS outbreak also started from China and then spread to even it spread as far as the US and spread to many countries of the world. But what was one problem with the SARS virus was that the infectivity was a bit low. It could not attach very well to the human cells, and so people who were and it was easier to control than this present COVID-19 because people who were infectious already exhibited the disease. So people were already sick when they were infected. And
there is no asymptomatic phase in this case of SARS.
Yeah.
That's the main advantage.
Yeah. I mean, asymptomatic people are not infectious. That was the main difference. So what happened is governments could control this disease earlier or easier compared to our present SARS-CoV-2 virus. Because the people who were infectious, they were exhibiting all the symptoms, so they could be quarantined easier, and the government could control the disease earlier. So that was the main difference in the SARS virus. So, but even then, when we were researching this topic, I found that by the end of 2003, around 750 people had died close to 750 due to this SARS pandemic. Okay, and another pandemic called MERS was also, it was a smaller pandemic that happened in 2012 and it was also a coronavirus. So next let's talk about PCR, polymerase
chain reaction. Actually, it was first developed by Carey Mullis, a famous American scientist, and this RT-PCR, reverse transcription polymerase chain reaction, is now considered as the gold standard for diagnosing COVID-19. So we will see first the basic principle of PCR test. PCR will amplify some parts of DNA or RNA, genetic material, and identifies the genomic sequence.
So basically, if there is a particular genetic sequence in a particular sample, it will make many copies of that particular genetic sequence, and we can find out whether that particular genetic sequence is present in that sample. It's like if there is a needle in a haystack, it will make many copies of that needle, and you can find that needle easier. So that is the basic
principle of PCR. In case of COVID-19, it will find out that particular genomic sequence and PCR positive. So, Dr. Andrew Kaufman, who is a famous American doctor and researcher, he has pointed out some defects in using PCR as a gold standard test for COVID-19. First of all, you all know the first outbreak of COVID-19 happened in Wuhan, China. And there the authorities in Wuhan first detected this virus as the cause of this COVID-19 infection. Then what they could have done as a second step is that they could have purified or isolated this virus from the infected patients and developed or identified the full genomic sequence of this virus. Before doing it fully, what they have done is that they have come upon PCR as a diagnostic test for this COVID-19 infection.
So even before they fully purified the virus and identified this virus as the cause of this disease, they hit upon PCR as the diagnostic test for this thing.
So this Andrew Kaufman points out that PCR can be positive in certain other conditions also, like infections with other coronaviruses, asthmatic conditions, then toxic injuries, ionizing radiations, lung cancer. In all these conditions also, PCR may be positive for these patients. So what is the drawback is that this PCR test has got high rate of false positivity, nearly 80%.
80%. So it's almost like 4 out of 5 cases can be falsely positive in PCR if you do the PCR test.
In COVID patients.
Yeah.
So we are also adding the link of his YouTube video in our description box.
You can watch these videos in detail, where is examining the PCR test and the problems with that. So next we'll look into a very interesting topic. That's event 201. What happened is that in October 2019 there was a meeting of some very high-powered organizations like the World Economic Forum, the Bill and Melinda Gates Foundation, and this happened in New York actually in October. So what they actually did was, there were several people present. There were many pharmaceutical companies, representatives of top governmental health bodies, international health bodies. So what they did, it was actually a pandemic simulation exercise. So what they did was, they were saying that suppose there is a hypothetical pandemic or an imaginary pandemic which is happening in the world. So how will be the response of the governmental bodies, how the pharmaceutical companies will respond, how the WHO will respond. They were trying to prepare these many organizations for response in the case of a worldwide pandemic. So, but interestingly, their scenario was that there is a coronavirus pandemic, and it actually broke out from some pig farms in Brazil. Pigs were infected from bats, and then pigs were infected, and then pigs infected human beings. And this whole disease spread over the world and became a pandemic. And it was also a respiratory disease in this imaginary scenario. It's called CAPS, Coronavirus Associated Pulmonary Syndrome. And and many people were actually infected, and actually in this hypothetical scenario, they said that it lasted for something like 1.5 years, and there were 65 million deaths in their imaginary pandemic. So you can see these videos, and when you see these videos, you will feel that like they are discussing all the strategies of how to deal with this pandemic and everything. And so what researchers find most very coincidental is that barely 2 months later, an actual pandemic of coronavirus broke out from China. And more like, there's another point, that this actual pandemic broke out, but still we did not seem to be prepared at all. The WHO, like first 3-4 months, it seemed like totally out of depths. It did not seem to realize that what was happening. It did not advise against travel to China. So you can actually watch these videos on YouTube. We'll be putting the link, and you can explore what is there in event 201. It's one of the controversial topics in this corona subject. And we'll also talk about a related controversy.
Controversy, that is an article published in journal Nature. Actually, COVID-19, is it natural or non-natural?
That is the main question.
So let's look at that article, which is published in 12th November 2015. And Nature is actually a world famous international medical and science journal.
So I will just give a bit of a background. They actually did a very controversial experiment on viruses in 2015 in the American University of North Carolina. And Nature, which is a very world respected international journal, it's describing this experiment. So we can go ahead.
So actually we all know, like as I told earlier, there are many coronaviruses primarily living in the bat population. So most of these viruses are not causing any infection or diseases in human beings. But there is a rare coronavirus called SCH014, which is the character of this virus is that it can attach easily with the human cells.
So it's like, unlike most bat coronaviruses, which cannot attach very well with the human cells, this particular virus can attach very well with humans.
But here the fortunate thing is that it cannot cause any infection or disease in man or human beings. So what they have done is that they have combined this SCH014 with SARS virus.
SARS is another coronavirus, like SCH014, but it's different.
SARS, the property of SARS is that it cannot attach easily with the human cell, as we told earlier. But once inside the body, they can cause high deadly diseases.
It's very pathogenic.
Very pathogenic. So what they have done is that they have fused the outer coat of this SCH014 with SARS virus. So and created a mutant virus. This mutant virus can easily enter the human beings, inside the body of human beings, and can cause deadly diseases because of the SARS inner core present.
So actually, they created a Frankenstein-like virus. Like the natural virus was not able to go inside the human cells, the natural SARS virus. So they actually fused this virus with another virus which can go inside. So what happened is, it's called as a gain of function. So the virus gained a character which was not there before. And so this deadly virus, after this experiment, it can go inside the human cell and it can cause deadly disease, this mutant virus. So,
and according to the opinion of two world famous virologists, one is Simon Hobson and other one Richard.
Simon Hobson is a world famous virologist from Paris.
And Richard Ebride from USA. The only result of this experiment is the creation of a non-natural risk artificially inside a lab. So also one interesting thing
I was reading about this thing, what Simon Hobson had said. He had said that once such a virus, they had created a non-natural risk inside a lab, totally unnecessary. And once such a virus, if supposedly it escapes from the lab, no one in the world can predict the trajectory of such a virus. What kind of damage it can do, no one can predict.
That kind of potential risk they have created. And in one more interesting thing here is that the Chinese virologist named Shi Zhengli, she was also part of this experiment. And she is actually the director of Wuhan Institute of Virology.
That's a very interesting point. That Shi Zhengli, the director of
Wuhan Institute of Virology,
she was part of this experiment which was led by Ralph Baric.
And one more thing, this COVID-19 outbreak happened in the same Wuhan. So is there any role for this mutant virus in the COVID-19 outbreak? That is a very controversial topic, whether that COVID-19 virus is actually the same virus which was produced in the lab in North Carolina. And I think many researchers are now looking into this topic. And also, another very famous Chinese virologist, Li Mengyan, she has recently said that she actually escaped from China because she feared the Chinese authorities, and she has escaped to the US. And now she is saying that COVID-19 is a bioweapon. She is saying it openly. So I think this particular angle of this experiment has to be studied in great detail. So actually, like when we look at all these events, the picture we are getting is that first there was an event 201, and so they seem to have some foreknowledge of this pandemic. And also there was this experiment in 2015 in which they have created a virus which is very similar to the virus which has actually caused that pandemic. So all these factors need to be looked into by researchers. And so presently our own country is facing a serious situation due to this. Around 3.2 to 3.5 lakh cases are coming. And also I think this particular year regarding this coronavirus, we are getting these mutant viruses, double mutant and triple mutant. UK mutant. These seem to be, and according to some doctor, like most of the doctors in the ICU, these particular viruses, they seem to be creating more severe disease. Like patients seem to be progressing to pneumonia faster. Still, like
So even if you get a COVID positive diagnosis, there is no need to panic. What we can do is there are some measures which you can take. Like if you are COVID positive and you don't have too many symptoms, you are staying at home, you can do some steps like you can do some exercise. Any kind of exercise like walking will boost the immune system.
Prone position.
Yeah. If you need to increase the oxygen levels, you can adopt a prone position. Like abdomen, you can take a pillow, you can put the abdomen on that pillow, and instantly the vital capacity of the lungs will expand. And the oxygenation level will go
Your
up.
belly should come over the pillow.
Yeah. And there is stuff like the pulse oximeter. Like you can put the finger inside the pulse oximeter and you can check your oxygen levels.
It's a small
it's a small instrument.
Actually.
It's a small machine. We have a pulse oximeter here. This is the machine actually. It will show the reading here. So once you put the finger inside this machine, it will show the reading over here, and you can instantly check the oxygen levels.
And if it comes below 95 or 94 like that, we have to be careful and you have to take hospital advice or something like that. That all things you can.
So we'll be coming out with the next video, and we'll examine these measures in detail, like what you can, if you get a COVID positive diagnosis, what are the small things which you can take care of to prevent you from getting into a serious situation that you land up in a hospital. So there are some stuff, and will be
also initial measures like SMS.
Right.
That all things we will tell in detail in that video, and it will be coming soon.
It will be coming soon. So our country has come out of bigger crises before. So we are a resilient people. So hopefully we will come out of this crisis. And recently there's a good news that our DRDO, Defense Research and Development Organization, has come out with a drug, 2-deoxyglucose, which is showing some good promise in this particular disease. And so hopefully we'll come out of this crisis in a few months. Okay, friends.
Stay home, stay safe.
Stay home, stay safe, right? And we'll be coming out with a video. So don't forget to subscribe us so you can get all these interesting videos on topics of interest. Okay. See you.
Thank you.